Hi, everyone. Welcome to OsteoBites. We're so happy that you were able to join us today. My name is Christina Iptoma, and I am mom to Osteo Angel Dillon and Director of Scientific Programs at MIB Agents. And today is a special episode of OsteoBites because we have not just one, but two guests today. Welcome to our dynamic duo, Dr. Matteo Trucco, a pediatric oncologist from Cleveland Clinic, and Dr. Kurt Weiss, an orthopedic oncologist from the University of Pittsburgh. They're going to be discussing their collaboration on a phase 1 clinical trial of Disulfiram with copper gluconate and liposomal doxorubicin in patients with treatment refractory sarcomas. Thank you so much, Dr. Trucco and Dr. Weiss, for joining us on OsteoBites today. We are thrilled to have you both, and welcome to Walker, who is our panelist today. Walker is an osteo warrior and vice president of our junior advisory board. So a little bit more about our guest today. Dr. Trucco is a pediatric oncologist and the clinical director of Pediatric Hematology and Oncology at Cleveland Clinic Children's, specializing in the care of children, teenagers, and young adults battling bone and soft tissue cancers. He also directs the Children's Cancer Innovative Therapy Program, where he and colleagues design, develop, and conduct clinical trials seeking more effective and less toxic treatments for childhood cancers. In addition to his roles at the Cleveland Clinic, he is co-chair of the National Pediatric Cancer Foundation's Sunshine Project Consortium, partnering with other top pediatric cancer centers to develop clinical trials. And he's honored to be on the board of MIB Agents, chairs its Scientific Advisory Board, and co-chairs the organizing committee for the Factor Conference. He also has the privilege of moderating the MIB Agents Tumor Review Board for Osteosarcoma, known as Turbo. Though honestly, Dr. Trucco, the privilege is all ours. And Dr. Weiss is an orthopedic oncologist and directs the Musculoskeletal Oncology Laboratory at the University of Pittsburgh, which is a basic science lab dedicated to the study of sarcomas. His mission is to help develop a world-class translational sarcoma research program at UPitt. As an osteosarcoma survivor himself, Dr. Weiss brings passion and enthusiasm to the laboratory, clinic, and operating room. Through the UPMC Hillman Cancer Center, he's also a proud collaborator with scientists who are trying to understand how other forms of cancer spread to and destroy the bone. Dr. Weiss is a founding member of the Musculoskeletal Oncology Research Initiative, MORI, the Pittsburgh Cure Sarcoma, the Pittsburgh Sarcoma Research Collaborative, and the Pittsburgh Center for Bone and Mineral Research. He's a peer reviewer for multiple journals, including the Journal of the American Academy of Orthopedic Surgeons, Biomed Central Cancer, Sarcoma, Cancer Research, International Journal of Cancer, and others. And he's a former member of the NIH's Center for Scientific Review Early Career Reviewer Program, and he served on multiple national Cancer Institute study sections. He's a member of the Musculoskeletal Tumor Society, for which he serves as chair of the research committee, and the Connective Tissue Oncology Society, for which he has served on the board of directors. So you can see they both have a lot of free time. So if you guys have anyone out there has any groups or organizations that they want to invite these guys to participate in, I'm sure they will love it. Because there's just not enough, I think, on their calendar. So as you can see, we're very fortunate to have both of them with us today. And just a couple of other announcements before we have them jump into their presentation. We have our monthly newsletter called Connective Issue, which goes out on the first Wednesday of each month. That means we have an issue going out next week. It's a monthly roundup of all things osteosarcoma, including recently published articles or papers, new clinical trials, job postings, funding opportunities, and spotlights on members of our community. And if you aren't already receiving it, you can sign up on our website, and I will put a link in the chat. We also have our bi-monthly virtual tumor board for osteosarcoma, affectionately called Turbo, which will meet again on November 8th. That will be our last meeting of 2023, and then we'll get 2024 dates on everyone's calendar soon. If you're a clinician or researcher with an interest in osteosarcoma, or you just want to spend more quality time with Dr. Trucco, our moderator, you're welcome to join. I know that's why Dr. Weiss joins Turbo. And so please, I'll put some more information in the chat, and please fill out the form if you'd like to join that. Walker, could you go ahead and introduce yourself, please?
My name is Walker. As Christina already said, I'm this year's Junior Advisory Board Vice President, and then also an osteo warrior, a little over 3 years in ED now. And I'm really excited to hear what our two presenters have to say today. So with that said, Dr. Trucco, you can take it from there.
Thank you, Walker, and thank you, Christina, for that amazing introduction. I'm not sure I deserve it. So we're going to try tag teaming here. Can you guys see the slides okay? Thumbs up. Yay. Okay, so I'm going to start off, then I'm going to hand it off to Kurt, then he'll hand it off back to me. We've presented this trial before. It feels like it's taken forever to get it open. But we'll kind of give each other's perspective on how it happened, the data behind it, and a little update on how the trial is going so far. Full disclosure, it's an active trial, so I can't tell you it's working, everybody enroll. That's not allowed. But we'll kind of tell you as much as can be shared along the way. So how this all started, I've been asked by trainees and other people, like, where do you come up with your ideas for clinical trials? And honestly, I was sitting in my office one day staring at a whiteboard, and I was like reminiscing on this paper that I wrote in fellowship with my mentor, Dave Loeb, on what are called sarcoma stem cells. And it's funny because back then when we wrote it, a lot of people kind of read it and was like, thought it was a joke. Like, there's no stem cells in cancers, let alone sarcomas. And nowadays, everyone kind of sort of accepts that there are these subtypes of cells that lead to this. That lab, Dave Loeb, published on Ewing sarcoma, where the population of cells that express this high level of this enzyme called aldehyde dehydrogenase, which you'll hear a lot about, seem to have properties of a cancer stem cell. So what is that? A cancer stem cell can recreate a new tumor. It's resistant to therapy. It kind of lays quiescent a lot of times, and it can grow in certain conditions that other cells don't seem able to. Similar findings have been shown in osteosarcoma and rhabdomyosarcoma, and those are the big three sarcomas we see in kids. So, this cancer stem cell model is like these small population of cells. They're in the tumor. They don't make a huge portion of it, but when you give chemo, you kind of wipe out the red cells here. The cancer stem cells are left, and then something triggers them, and they kind of grow and reconstitute a tumor. Another way of looking at it is you give therapy, and what's left is a multi-drug resistant cell, and that gives rise to a tumor. Doesn't really matter. Either way, there's clearly, when a relapse happens, cells that are resistant to that upfront therapy, and those are the ones that we have to eliminate if we want to improve cures. So looking at aldehyde dehydrogenase, not only is it an enzyme, not only is it a marker of this kind of resistant population, but enzymes do things. And aldehyde dehydrogenase specifically breaks down substances, specifically aldehydes. And there's more papers suggesting that not only does that enzyme identify that population, but it may play an active role in conferring that resistance. And so these are just a few papers that I came across when I was trying to see, well, what role was aldehyde dehydrogenase doing beyond just being a marker? And this name kept coming up. But at the time, I wasn't really paying attention. This is just a list of some of the chemotherapies that are affected by aldehyde dehydrogenase. And this is like a who's who of chemotherapy we use for sarcomas, including osteosarcoma. So one study we published, it took a long time to publish this, but I worked on this during my fellowship, again, with Dave, is trying to target those cells using a drug called temsirolimus, which blocks a pathway called mTOR. And we combined it with chemotherapy and trying to see if that temsirolimus would sensitize those resistant cells to chemotherapy. And then actually came up across this paper that rapamycin is in the same class as temsirolimus, and it seems to actually have a role blocking aldehyde dehydrogenase. And so maybe that's how temsirolimus was having this effect on killing that population. And again, this name keeps popping up. So the idea being, in the human, aldehyde dehydrogenase, actually the biggest role we know of is it blocks the conversion, it breaks down ethanol into a substance that can be disposed of by the body. And so you go out to a party, you have some drinks, aldehyde dehydrogenase is what's going to help you eliminate the alcohol. People that lack that enzyme inherently are the ones that kind of get bright red whenever they get a drink. It happens with some frequency in the Asian populations. And it turns out we actually have a drug that blocks this. And the drug is called Disulfiram. And it's actually, the brand name is Antabuse. It's a drug used to treat alcoholism where you take this pill every single day. It doesn't bother you until you try to take a drink, and then you get violently ill. And so the idea is it deters an alcoholic from drinking because they'll get sick. It doesn't work perfectly because the alcoholic has to be motivated to take this pill every day. But that being said, we have a 60 to 70-year history of using this drug safely and effectively. And so instead of using rapamycin to try to, attack aldehyde dehydrogenase indirectly, we have a great drug that hits aldehyde dehydrogenase directly. And there's a bunch of theories of why aldehyde dehydrogenase is elevated and why blocking it might lead to being toxic to cells themselves, but also potentially potentiating the chemotherapy. And again, there's a lot that's speculated about disulfiram, not just those pathways that I showed. There is no shortage of studies studying disulfiram in cancer. And so it seemed like, okay, let's go ahead. Let's work with this. And then specifically in osteosarcoma, it's been shown that disulfiram itself has some activity in preventing metastases and blocking the growth of osteosarcoma. And there's that name again. And now I will turn it over to that name, Dr. Kurt Weiss, who did a lot of that preclinical data that supported my study. And I'll let him talk about that. And then we'll talk about the study, the trial later.
Hey, everybody. I'm Kurt, and I'm going to take over the story for a little bit. Christina, thanks again for having me. It's always an honor. You guys don't need any more information on osteosarcoma, except for maybe one little thing here, which is that just about all metastases are to the lungs. And this is what keeps me and Matteo up at night, is that, as he said in a few slides ago, the cells that die with chemotherapy or die because I take them out with my knife, those don't bother him and they don't bother me because they're looked after. It's the cells that are making it past my knife and past his chemotherapy that are going to the lungs and causing trouble. That's what we got to figure out a better way to fight. So I'm pretty focused on this idea of doing better with metastatic biology and metastatic disease. So this is the good news, good news slide, or bad news slide. Before Matteo and his colleagues started giving chemotherapy to kids with osteosarcoma, we did terrible, between 10 and 20% survival with surgery alone. Well, when we became audacious enough to give children and adolescents and young adults chemotherapy, we did a whole lot better. And that's the great news. Chemotherapy is, osteosarcoma is one of the great success stories of chemotherapy in modern times. But that's where the good news stops, because we've been at 65, 70% 5-year survival for most of my lifetime. And the more bad news is that we do just as crappy for kids with metastatic disease as we always did. So there is room for growth. So the young lady on the right there, that's Katie. Katie just came into my clinic on Tuesday with her diploma from West Virginia, and she's going to go to graduate school and be a basic scientist and do oncology research. But when I diagnosed her 26 years after I was diagnosed, our prognoses were the same. Now, I certainly hope that there's nobody that thinks that's okay. All right. So metastases, bad. Metastases is the big problem. So wouldn't it be cool if we had 2 cell lines that were alike in all things, except one was really metastatic and one wasn't very metastatic? That would help us figure out the factors that confer metastatic potential. Okay. And it turns out that exists. And this is the model I learned from Dr. Hellman and Dr. Khanna at the NCI when I was with them as a medical student. So the K12 mouse osteosarcoma cells, they'll metastasize, but they kind of putz along, whereas K7M2 is violently metastatic to the lungs, and pretty reliably so. So when you hear or see the M in K7M2, think metastasis, because that's what that guy does. Okay, and Matteo talked about ALDH and why that's important. It's a stem cell and a cancer stem cell factor. A lot of literature in cancer, just like Matteo pointed out. But specific for osteosarcoma is, did I put Kanya? Yeah. So this last paper is by my buddy, Dr. Kanya Hinoki, who's actually in Nara, Japan. And he suggested that high ALDH activity might lead to chemo resistance, which is one of the things that piqued my interest in ALDH. So we did a very crude experiment. Dr. Mu basically poured some peroxide on cells, the K7M2 and the K12 cells, to see what would happen. And it turns out the K7M2s were more resistant to that oxidative stress. Okay, well, might this ALDH thing be the reason for that? And it turns out that the K7M2 cells were making more of the gene for ALDH, and also they had greater ALDH activity on this assay. So that green hump there, that shows you the ALDH activity. And this has a big green hump, and the K12 has an itty bitty green hump. So we know that their activity for that enzyme is different because, like Matteo said, enzymes do stuff. And that's why we're interested. So we separated the K7M2 cells into the high ALDH expressors and the low ALDH expressors. And it turns out that they not only look different, but they act different. When we ask them to invade through a three-dimensional matrix, the ones that have high ALDH are real good at it, and the ones with low ALDH are not so good. Okay, so what do we got sitting around the lab that inhibits disulfiram? Inhibits ALDH, and Matteo already gave us a great introduction on disulfiram, what it does, why it does it, and so I thought, okay, well, let's put this on the cells and see what happens. Well, they don't like it too much. And this is all terrific, but the last time I checked, there were no mice walking into Matteo's office. And to this point, all of the work we had done is on mouse cells in a dish. So I thought, well, if we really think this is important, let's see what it does in our human sarcoma cells. So we have a tumor registry and tissue bank where we're able to keep some of the tissues from our osteo warriors who graciously donate them and take them to the laboratory and study their biology. And on the left is a chondrosarcoma, which is a cartilage cancer, and on the right is an osteosarcoma. And that's what it looks like in the dish. And this is from one of our patients. So we took, this guy's in your neck of the woods, Matteo. Nikki's at OSU. And does joint replacement there. But when Nikki was in my lab as a resident, he looked at 10 consecutive bone sarcomas to see what their ALDH activity was. Now, we knew from their clinical records if they metastasized or not. And so we said, okay, does the ALDH activities in these cells have anything to do with whether they metastasize or not? And what happens if we pour some disulfiram on those cells? Well, the results were pretty interesting, and Matteo already cited this paper. We saw a pretty good correlation between the clinical event of metastasis and the ALDH activity in those patients. So at the bottom, again, that big green hump, this is a patient who metastasized, and this is a patient who didn't. With that, you can hardly even see that green hump there. And when we poured disulfiram on their cells, they didn't like it very much. So, okay, nobody's going to give us a Nobel Prize for a study of 10 patients, but, you know, it's maybe we're going in the right direction, especially for diseases like chondrosarcoma. You know, look, osteosarcoma is awful, but at least we have some weapons. Chondrosarcoma, there are no weapons. So we went back to the animal model, and Jared, when he was in the lab, he's a big shot spine surgeon in Dayton now, and when Jared was in the lab, he went back to the animal model and compared disulfiram and doxorubicin head-to-head in our mouse model of metastatic osteosarcoma. And I had a lot of slides on the different things we looked at and all these things, but it was a little bit too geeky even for me. So let's just cut to the chase and show the important slides, which was that there was no statistically significant difference in metastases between the doxorubicin and the disulfiram animals, and they both did better to the control group who just got salt water. Okay, so here were our conclusions, again, to cut to the chase. Disulfiram was equivalent to doxorubicin in terms of its ability to limit metastasis. It outperformed, and we have that in air quotes because this is based on how we think cancer cells work, outperformed doxorubicin with things like mTOR inhibition that Matteo talked about before and some other things that we think it's better on a molecular level. And finally, if you look at the side effects compared to doxorubicin, they're not so bad. And also it's, pennies on the dollar cheaper than doxorubicin and much safer. We've some of the people on this this webinar have taken doxorubicin. It's no fun. It beats your heart up. It's rotten. And if there were a way we could get away from doxorubicin-based treatment with the same or better results, that would be a pretty good thing. And I won't steal all of Matteo's thunder just yet. Okay, so then we said, all right, well, again, we're a little bit heavy on the animal models. So let's see if we can find something to the K7M2, K12 story, but in human cells. And it turns out my scientific mom, Jeannie Kleinerman, did this. So she took SALS2, which is kind of a very old workhorse osteosarcoma human cell line, and she cycled it through the lungs of experimental animals to get these really angry big brothers of SALS2, which she called LM2 and LM7. And I said, okay, well, if we think ALDH is important, and we do, then the ALDH in LM2 and LM7 ought to be more than in SALS2. And it turns out it was, which is nice. We expected this, but until you prove it, you didn't prove it. So that was cool. And then, so this was previously unpublished stuff, but it's going to be coming out soon. Again, I'm pretty obsessive about this whole metastasis primary tumor differences thing. So I wanted to look at RNA sequencing between primary osteosarcomas and metastases, and in some case matched sets of those two. And because some pretty smart people, including my bosses down at the NCI, said, Kurt, you're wasting your time. You look at the cell in the femur and the cell in the lungs, you're going to see apples and apples. They're the same. Osteosarcoma is a metastatic tumor from the very first cell. Now, I don't believe that. I never believed that. But until you prove it, you didn't prove it. Bernie Fisher always said, in God we trust, but everybody else needs to bring data. So I needed to find some data to bring. Okay, so this slide shows us that the genetically, the metastases and the primary tumors are different animals. They're different tumors genetically. And so now we've shown it, and now I'm happy. But what I did, and in fact, metastatic tumors have more in common with other metastases than they do with their own primary tumors, which is absolutely fascinating. In any case, so now I've proved it, and I'm happy. What I didn't expect to see was that ALDH expression was higher in the metastasis than in the primary tumors. Now, obviously, when I saw those data, that was like Christmas, my birthday, and the 4th of July all at the same time. Because these are what's called unbiased data, meaning you put your stuff in and you get an answer. There's no way to tinker with it, right? You get what you get. And so this was really, really powerful that without looking for ALDH, we found ALDH. Okay, so once again, ALDH reared its ugly head, which made us very, very exciting. But we were a little bit silly because we didn't bring our lucky penny. Now, what does that mean? Well, copper is an essential micronutrient that helps a lot of these enzymes Matteo was talking about, helps them do their thing. Oh, okay, well, who cares? Well, copper metabolism seems to be all banged up in a lot of different cancer types, and we think that copper might be important to the growth of cancer cells. Well, why do we care? Well, we better care because copper and disulfiram seem to have this weird synergy going on, which I didn't know anything about until my PhD student, John, pointed it out to me. So we were like, all right, well, maybe if disulfiram on the cancer cells is good, disulfiram and copper is even better. So what are K7M2 and K12 doing in terms of copper? Oh, snap. This isn't what we expected to see. K7M2 is super, super high and super, super low, and K12 is super high. Well, that's not what we expected to see. And this holds up over time when we treat the cells. The K12 cells just can't get enough copper, and the K7M2 cells don't like it very much. So isn't that weird? Well, then we looked at the enzymes that control copper metabolism. CTR1 is the major copper sucking in enzyme, and ATP7A is the main copper pushing out enzyme. So it looks like the less metastatic cells are gobbling up the copper, and the more metastatic ones are sort of pushing it out into their microenvironment, which is very interesting. So when we looked at our K7M2 cells, lo and behold, when we put copper on them plus disulfiram, they didn't like it very much. All the cells died. And we think we found a sweet spot where we could kill the K7M2 cells and spare normal cells. These 3T3 cells, they're normal fibroblasts, skin cells. So, anybody can pour a chemical into a dish and kill cancer cells. That's not hard. If you pour chemical into a dish and you kill cancer cells, but you spare normal cells, well, now you got my attention. That's cool. So we treated cells with disulfiram and copper, and again, the K7M2 cells, they didn't care for it very much. But they always seem to recover. Now, we noticed from earlier studies that when we used disulfiram on the cells and we used doxorubicin, that different genes went up and down. So we thought, well, if these two chemicals are doing the same thing, but in different ways, maybe that suggests that we can use them together. Maybe we can combine that treatment. And so I think this is my last slide, and it's also my favorite slide. So you treat cells with doxorubicin, that's this red line here, and you kill them, but then they come back. You treat them with some disulfiram, and you kill them, but they come back. You treat them with doxorubicin, disulfiram, and copper, they died, and they stayed dead. So I think this is one of the pictures that from the paper that we wrote that Matteo really liked the looks of. And so he saw these data, and we talked to each other, and he said, Kurt, maybe it's time to take this out for a spin and see what it can do. And we were going like gangbusters, and then the pandemic happened. But at that point, I think I'll turn it over to my much smarter, much more handsome friend to tell us the rest of the story.
That friend couldn't make it, so you're back to listening to me. With all that great data that Kurt shared, we decided let's go ahead and try this, see if we can develop a clinical trial. And anytime you try a new combination, you got to establish safety first and foremost, and then you try to figure out efficacy. And what are you going to combine it with? That's a question that came up a lot. Originally I designed this, what we call a 3+3 dose escalation trial, where you enroll 3 patients at 1 dose. If it's not too toxic for those patients, then you can give a little bit more to 3 other patients, and you can give a little bit more to 3 other patients. I'm a pediatric oncologist. I wanted to include younger patients. Start with kind of teenagers, but then ultimately get younger patients. While Kurt's data is an osteosarcoma, there are parallels in other sarcomas, and the poor prognosis and failure to improve therapies actually is present in other sarcomas too. So we kind of wanted to make it applicable to more than one sarcoma. And the aldehyde dehydrogenase story seems to be true. Anyway, And to better understand exactly what's happening in a human compared to what's happening in a mouse or cell lines in Kurt's lab, we want to get tumor biopsies along the way. And obviously, a patient needed to abstain from alcohol. So this actually went through various iterations, in thinking, okay, when a patient relapses with osteosarcoma, what do we treat them with? And then again, because I was thinking a little more broadly, when a sarcoma relapses, what do we normally treat them with? Gemcitabine docetaxel is a common regimen used for various relapse sarcomas. And so originally I developed it that way, with that chemotherapy as a backbone. And actually I hadn't heard the whole copper story at that time. And so this was how I originally kind of designed the trial. Turns out docetaxel actually contains alcohol in it, and the one version of it that doesn't contain alcohol was off market at the time. So much for that. When I got the Outsmarting Osteosarcoma grant from MIB, it was originally with this chemotherapy backbone. Pete Anderson, who I share an office with, actually said, "You got a great idea here, but you have the wrong chemo." And he actually suggested a continuous infusion ifosfamide as the backbone. And so we kind of pivoted to that. Then Kurt's data came out, and the issue with ifosfamide is there's certain other sarcomas that ifosfamide's kind of used up front, and you don't always come back to that. So then the other thinking is, okay, ultimately we want to bring a drug like Disulfiram up front to give it with something like MAP chemotherapy if we think it's effective, because you want to get rid of those resistant metastasis potential cells get rid of them from the get-go so that patients don't relapse. So this is what we ultimately settled on, actually 28 days, not 21. But liposomal doxorubicin, it's a version of doxorubicin that it's packaged in little fat globules so that it's not as toxic. It doesn't hurt the heart as much. It seems to focus on, it accumulates in the tumor. And again, that data about copper came into play. And the way we designed the trial is, we'd screen patients, we'd get some biopsies, just core needle biopsies of their tumor, ship those to Kurt, and then we'll do this lead-in week. So you'll take the Disulfiram, and we had it, if you take copper and Disulfiram at the same time, it turns into this weird sludge. So we actually have them take the Disulfiram in the morning and the copper in the evening. And then we do that for a week. Then we start giving the Doxil. Actually, it's monthly, not every 21 days. And after 2 cycles, repeat scans, see if it's shrinking the tumors or at least keeping them stable, and get another core biopsy so that Kurt can investigate what's actually happening in the tumor after being exposed to Disulfiram, copper, and liposomal doxorubicin for 2 months. And then continue this for as many as 12 months to see how the patient does, repeating scans every 2 months. At that point, we're also enabling patients to get radiation if they want radiation, have surgery if they want surgery, and just holding the chemo so that they can safely have those interventions. So it took a couple changes. The FDA insisted that I only administer this to adults first before seeing if it was safe enough to try it in kids, because guess what? There's not a lot of kids running around with alcoholism, so there's virtually no data for Disulfiram safety in the pediatric population. Anything you get when you Google pediatrics and Disulfiram is little Johnny got into mom's Disulfiram and took the whole bottle thinking it was candy, and he got a massive overdose and toxicities from that. So the FDA insisted I treat adult patients. The study's still designed for relapsed sarcomas, adequate organ function. They still need to be able to take pills. And since, you wear a hat of a researcher and a physician, you want to do what's best for the patient, but also best for the trial. The biopsies, I think, are essential to understanding this, but at the same time, we don't want to put patients through a risky procedure needlessly. So we actually have it built in to the trial that if a biopsy is deemed risky or dangerous, we'll waive that for a patient. So for example, I have a patient who has one nice relapse metastasis right next to his heart, about a centimeter large. We're not going to have an interventional radiologist try to stick a needle through the man's chest, through their lung, into this 1 centimeter tumor that's right next to the heart. That's just too dangerous. And so we've waived that biopsy requirement for certain patients. Patients must abstain from alcohol, and there's actually a list of other medications that's just not safe to give with Disulfiram. So patients have to avoid those. Interestingly, this has come up probably more than alcohol, is marijuana and its derivatives actually is broken down by the aldehyde dehydrogenase enzyme. And so they have to abstain from those compounds as well. And again, since we don't have a clear understanding of how Disulfiram works, it may be blocking aldehyde dehydrogenase, it may be transport of copper, it may be something else. There's plenty of things that it does. That's why we're trying to get those biopsies. We're also drawing copper levels and sending those, measuring those to see what the copper level does during these treatments. A big question that comes up is if Disulfiram increases the toxicity of the chemo to the cancer cells, won't it increase the toxicity to normal cells? Again, fortunately, this drug's been around for a long time, and it was actually being tested to see if it could be protective against a lot of chemotherapies. And back in the day, I forget how old this paper is. I think it might be from the 80s. They actually tested it in combination with cyclophosphamide, which is an aldehyde, which is broken down by aldehyde dehydrogenase. And despite that, when you combine it with Disulfiram, there's actually no increased toxicity in the bone marrow. So it doesn't seem to have any extra effects on the bone marrow. Here, there's actually a couple papers, mostly in mice, showing that it might protect against that toxicity that Kurt referred to in the heart from doxorubicin, might even protect the kidneys from some of the toxicities from ifosfamide. So again, this made us feel like, okay, it should be relatively safe to combine these drugs in humans, but you never know. Disulfiram's been around for a while. Teva is actually one of the largest generic drug manufacturers. I reached out to them. I said, could you please give me some Disulfiram for my study? And they said, we're not going to make a buck on this. Of course not. That's when a group called Cantex Pharmaceutical actually kindly reached out to us. Steve Marcus, that was actually mentioned in Kurt's title slide, called me, emailed me out of the blue, having heard that I was talking about developing this trial, and said, we make Disulfiram. We've been testing it in cancers. Would love to, partner with you and provide you the drug. And I said, awesome, that's amazing. We got to get my buddy Kurt on board too. And he's like, that was my next call. I was like, perfect. And so in the meantime, I prostituted myself out to try to get money to support this trial. Fortunately, Outsmarting Osteosarcoma and an internal grant called Velasano came through to support us.
I'm very glad we saved you from that. I've never heard that one before, so.
Yeah, well, that's what you got to do sometimes. So and then COVID hit, and it's been a disaster trying to get it open. finally, January 2023, the trial opened, late 2023. Dr. Anderson sees a ton of patients, and he's been promising this trial to patients for years now. So we were finally able to start enrolling patients. We screened some. The first patient started treatment March 2023, and I gotta say a huge thank you and appreciation to them. Like, I literally had to tell this guy, "You're the first human being that we've put these 3 drugs into at the same time. I have no idea what's going to happen." As the chemo infusion was starting, I poked my head in his room and asked him if he was turning blue or growing a second head, and he assured me he wasn't. But it's not. It doesn't escape me how much faith they put in us as physicians to try these new combinations. So to date, we actually screened 6 patients. Again, we can only treat 3 patients at a time. We screened 6 of them. We actually have to measure a copper level to make sure patients don't have any problems metabolizing copper, because if you have a problem metabolizing it, you can have toxic levels of copper. One patient, his copper level was just too high, so we didn't think it was safe. So he screen failed. Currently, 5 patients have been treated. Unfortunately, and it's a balance of clinical trials, you want to make it available to as many patients who might derive benefit as possible, but you also want to, ideally have the perfect candidate. The perfect candidate is someone who just relapsed, never saw any of these drugs before, has very small metastases, but they're measurable reliably, has no other side effects or symptoms. And yeah, that's great, but I don't know any patients like that, quite frankly. So, you open it up to any relapse patient with measurable disease. And we actually had it such that you can have had doxorubicin and liposomal doxorubicin before, because the whole premise here is that this combination sensitizes you to those drugs. Maybe that's asking too much of this combination, but we did allow patients who had received the chemotherapy before, especially for this first phase, was just trying to figure out the safety. So of the 5 patients that were treated, 2 of them actually had to come off the study early because they actually developed progression, quite frankly, before the drug even had a chance to work, I think. But these were patients that had tumor kind of everywhere. 2 patients did make it to the end of cycle 2, where we could, evaluate things again. Wish I could tell you there have been some home runs. So far, no. Again, we're at 1 dose level. We're going to bump it up, but it's still early on, and thus far, we haven't had a patient who has not ever been treated with doxorubicin before. So we are collecting tumor samples. We're basically batching them all here, and we'll send them to Kurt, and we'll save on the FedExing and just send it all together. So again, the analyses can kind of all be done together, but they're being stored right now. Again, 2 patients, we thought the biopsies were too risky. We do have what's called pharmacokinetics, so measuring drug levels. Again, we don't know what effect the doxorubicin has on the levels of disulfiram within the blood. And we're also collecting copper levels. Generally speaking, though, nobody turned blue. No one grew a second head. Pretty well tolerated. The most common side effects we heard about is some interesting dreams, which weren't expecting that one, and rashes. Blood counts don't seem to go too far down. Heart function has been okay, and nothing's limited our dose. And so we just started our hopefully last person on the first dose level yesterday. If they do well for a couple months, without any toxicities, we'll be able to bump up the dose and open it up to another 3 patients. So, so far, encouraging. Again, no home runs. Not going to pretend that we've seen dramatic responses, but so far safe. And again, a huge thank you to everyone involved. Kurt's been fantastic throughout this. Stephen Marcus, again, really came through providing drug, so we didn't have to buy it. My old mentor, Dave Loeb, supported me throughout all of this. Damon Reed has been an incredible supporter of this concept and offering suggestions along the way. And everybody here at Cleveland Clinic, Pete referring patients, Belinda and Reglan are doing the pharmacokinetics, our nurses, Melissa, Shauna, Alyssa, and Tina, getting this trial up and running and coordinating things for our patients. And the funding, again, the ones in the middle are Kurt's, the ones on the outside are mine. Again, we can't do this without funding, and it's great that people see the potential in this and are willing to support it. And with that, I'll open it up to any questions. Again, it's been a great collaboration across the Pennsylvania-Ohio Turnpike. We promised to have dinner and a beer together, maybe when the trial's over. With that, I'll stop.
Sounds like a plan on the beer after the trial's over. Thank you both so much. That was so interesting, and also just fabulous collaboration in terms of just getting this going, and having Dr. Weiss's lab do the correlative studies on this. So one question I had, and everyone, please feel free to put some questions in the Q&A. And actually, Walker, do you want to start with the first one?
Yeah, I'll start with the first question. They were asking, how long do you foresee the dosage safety phase study going? And then how long would the next phase for efficacy go?
Yeah, so the way we do clinical trials is the phase 1 trial is what's the dose? What's the safe dose? Phase 2 is where you really tackle efficacy. Phase 3 is where you compare the new treatment addition of a drug like Disulfiram to the standard of care and see if it actually improves things. So we're in the phase 1 world right now. We have 2 dose levels built into this trial. Obviously, I want to know what the dose is, but I really want to know if it works. And so we're still looking during the dose escalation at efficacy. That's why we need measurable disease. That's why we still evaluate. And so 3 patients in each dose level. So once, again, the current patient, if he finishes the 2 cycles and it's considered safe, we can go to the next dose level. At that point, I'm honestly going to reach out to the FDA and say, I realize it wasn't exactly what you guys had in mind, but we did technically treat, a lot of patients. Can we open it up to kids? And so we'd have to amend the protocol. If there's a signal, if there's something that suggests this might work, and maybe Kurt can figure out why it works in some patients and not in others, then it would probably be a whole new trial. Sometimes you can, convince the FDA to let you expand the trial to continue. And essentially we'd try to figure out the quickest, safest way to do it. One of the limiting factors is right now it's open at Cleveland Clinic, and with a couple grants that I threw together, we see a ton of patients. That's true, but while in a phase 1 trial, you can get by with 6 or 12 patients and answer the safety question. For an efficacy question, you really need close to, a few dozen, close to 50 patients, potentially. So you just need to kind of cast a wider net. Again, whether that's through a consortium, through, partnership with actually treating patients at both Cleveland Clinic and Pittsburgh, or something else, we'll cross that bridge when we get to it to see how we can move this forward.
I want to sneak in a question about, I'm kind of thinking about Dr. Weiss's slide that kind of showed the difference between the primary tumors and the metastatic tumors and the levels of ALDH. But I'm also kind of thinking about, there are some tumors that are seemingly chemo resistant, primary tumors that are resistant to chemo, right? Like they just have low necrosis. And then you have some tumors that seem pretty responsive to first-line chemo, high necrosis, but yet then there are probably some of those, cancer stem cells or the drug-resistant cells that somehow survive and then come back and flourish. And so I'm trying to wrap my head around, is there a difference between those two types of chemo resistance? So the primary tumor cells that seem to be chemo resistant, are they different from these, metastatic cells that are chemo resistant?
Matteo, what do you think? I got my answer. What do you think?
There could be a difference. We just don't know. It'd be great to look. But yeah, I don't have a great answer because those are three different beasts. I think they behave somewhat differently. I'd also throw in, the patients that have wonderful necrosis, everything's great, everybody's high-fiving each other, and then they relapse. Like, what the heck happened there? I don't know that aldehyde dehydrogenase is the only explanation. But again, it could be. We'd just have to look. Kurt probably has a much more eloquent answer.
Yeah, probably not. So aldehyde dehydrogenase is certainly not the whole answer. But to your first question, Christina, is a fascinating one, and my answer is so the question was, are the resistant cells the same as the metastatic cells, or are those two different niches? I don't think they are. I think you've got some cells in the tumor that are real good at pumping out the chemotherapy and have induction of a gene called MDR, multi-drug resistance, which P glycoprotein has about 5 different names. It's all the same enzyme that make them resistant. And I think they're the same ones that are resistant to stress and have that ability to metastasize. That's a guess. I think it's an educated guess, maybe, but I do think here's what I know for sure. Not every cell in the tumor is created equal, right? And nor do we have to kill all the cells in the tumor. We must kill the cells that are able to do those two things that you brought up, those essential functions, metastasis and drug resistance. And I think they're probably the same cells. That's a guess.
Thank you.
There's another question in the chat that says, can you share any ideas on other combinations with Disulfiram that should be tested for targeting metastatic progression? And I guess I would add on to that, would you guys try other chemotherapies like methotrexate and cisplatin? And then would you also try like anything besides copper, like other elements that are typically found in the body?
Certainly would be willing to. It requires a whole different trial or a huge amendment to the trial. And, we gravitated towards doxorubicin just because we had some data in osteosarcoma. We don't have a ton of data with some of those other compounds other than, oh, look, high levels of aldehyde dehydrogenase seem to have an effect on that laundry list that I showed. But again, the data is a little sparse. The cyclophosphamide, ifosfamide, certainly there's a more kind of direct path because we know those compounds are broken down by aldehyde dehydrogenase, and we know that if you expose cells to those drugs or tumors to those drugs, that enzyme goes up in response to that exposure. So there's certainly potential for all of those. There's just not a ton of data to kind of support which one to go after next. And you asked another question. What was your question on top of?
I was just asking on top of that about, I guess you kind of covered other chemotherapy. So I was asking about anything besides copper that you guys would try.
Oh, copper.
So there's this whole area of research called pheroptosis, which is apoptosis or cell death that is sort of brought about by iron. And a lot of the heavy ion transporters take both copper and iron and other things. So I think the answer to both your questions, Walker, is yes. Yes, we would like to try it with other drugs. Yes, we would like to try it with other heavy metal ions, but I think you got to start somewhere. And this is where Matteo and I are starting. And God willing, there will come a day, maybe not in our lifetimes, that we can answer all the questions you just asked. Okay, how do we get these? Because really what you want to know, what we all want to know is, how do we get these things to work at their best, right? What's the best heavy metal to combine with Disulfiram? What's the best dose of that? What's the best chemotherapy to put it with? What's the best? And I don't know that we're going to get there any other way than some significant trial and error. Matteo, your thoughts on that?
The copper comes from the active metabolite. So you take Disulfiram, it completely, disintegrates once it hits your stomach, and it turns into an active metabolite, which immediately binds copper. And in fact, Stephen Marcus loves showing this picture of what happens when you combine Disulfiram and copper in a beaker. This is disgusting sludge. So again, that happens, and the active compound that blocks aldehyde dehydrogenase is this Disulfiram compound bound to copper, which is why not just in our study, but most other studies testing Disulfiram in cancer uses copper. But again, like Kurt alluded to, there's other stuff going on, and we don't know that same thing that blocks aldehyde dehydrogenase and alcoholism is exactly what the active agent is in an anti-cancer role. So yes, would love to explore all that.
So unfortunately, we're already at the top of the hour. That went by really quickly, but you guys are so fun. So I knew the hour would go by quickly. If you have any questions, if you want to just put them in the Q&A, and we can always pass those on and respond to them, and pass them on to Dr. Weiss and Dr. Trucco and see if they can respond in email. But thanks so much for joining. And I just a quick rapid fire, just because people who might be interested in enrolling in the trial. So washout period?
Washout, I like to keep things relatively loose. So it's like the toxicities of prior therapies have resolved. So give it like 14 days. Your counts need to be back to normal, stuff like that. And okay. In fact, we had a patient just have radiation relatively shortly before.
Okay, I was just going to ask that because I remember you mentioned radiation and surgery. Okay, but I couldn't remember the time frame. So radiation slightly before was okay. And then What about when they're on the trial?
When they're on the trial, we ask that just stick with this for the first 2 cycles so we can evaluate safety and efficacy, because we're doing scans at the end of cycle 2. After that, again, my doctor hat takes over and says, let's do what's best for the patient. So again, osteosarcoma, surgically removing the tumors is the standard of care at some point. So after we get that 2-month point, we got to time it around the chemotherapy when it's safe. Whether or not we continue the Disulfiram while they're recovering from surgery is up for debate, but it's built into the protocol to have some leniency there.
Great. Thank you so much. So after 2 cycles, thank you both, Dr. Trucco and Dr. Weiss, for joining us on OsteoBites today and for making it better for osteosarcoma patients. More information on this and all OsteoBites can be found on the MIB Agents YouTube channel, on our website at mibagents.org, and at your favorite podcast place. And next Thursday, we're going to be joined by Jim Palma from the Target Cancer Foundation, and he's going to be discussing TCF-001 TRACK, which stands for Target Rare Cancer Knowledge, a clinical trial studying precision medicine in rare cancers. So tune in to learn more about that. You can find our OsteoBites lineup for the next 2 months on our website. And if you have any ideas for future topics you'd like to hear about, please share them with us at events@mibagents.org. Thank you again to Dr. Trucco, Dr. Weiss, and Walker, and for all of you for spending an hour with us today. And we hope to see you back here on OsteoBites when we talk to Jim Palma from the Target Cancer Foundation. Thanks, everyone.